Good afternoon. To start today, I want to ask you, when did you first get an interest in neuroscience?
Um, Neuroscience specifically, not until I was an undergrad.
Mm-hmm.
But I will say I went to a high school in a more rural area.
Mm-hmm.
And so I didn't have a lot of exposure to science or science-based careers. So it
Mm-hmm.
wasn't until I went to college that I really understood that I could pursue neuroscience as
Mm-hmm.
a major. And I was always really fascinated by how intricate the human brain was. And I was a lot more interested in that than like ecology and evolution. And
Mm-hmm.
I was a lot more interested in like physiological aspects of science, more like anatomy and physiology, pharmacology,
Mm-hmm.
even human psychology. And so that's why I think I was more interested in the neuroscience.
Mm-hmm. Yeah, I agree. I think that if you are raised in like a more rural area, it can definitely be a lot harder to get a lot of more exposure.
Mhm.
I do see that you are a postdoc researcher at UT Southwest.
Mhm.
Have you had any like unexpected encounters in your research so far, and did it like affect your work, or has your work mostly been smooth running and linear to what you've been doing?
So it's funny that you ask that. Actually, I had much more difficult time during graduate school than I'm having now as a postdoc.
Hmm.
So science is almost never a nice, linear, smooth process.
Hmm.
Um,
Right. Yeah.
a lot of the times we'll publish a paper and the paper is really nice and there's a well-rounded story, but getting to that is often unexpected. Like we have a lot of unexpected results because what you want to do as a scientist ideally is follow the data,
Hmm.
not kind of direct things the way that you want them to go. So in
Hmm.
graduate school, I had a more difficult time, mainly because of I was working with zebrafish at the time,
Hmm.
and we had some problems with the health of the animals, and that always makes research incredibly hard. If you don't have
Hmm.
healthy animals, and you're doing animal-based research, um,
Hmm.
that can stall things. And so I
Hmm.
had more of a problem with that in graduate school. Um, I have had like unexpected results. So my work is currently focused on sex differences
Right.
in Fragile X syndrome,
Hmm.
um, and specifically related to estrogen receptors within the brain. And so
Right.
we have been surprised by some of the findings, but not,
Hmm.
I wouldn't say that they've been disappointing, just maybe unexpected.
Hmm. Um, have you had any really unexpected trajectories, or is it just mostly like a rough experience at like your master's degree?
So actually, I had more trouble. So I didn't get a master's degree. I just went from undergrad. I had 3 gap years where I did research. I
Hmm.
was actually doing research here at UT Southwestern. I was a lab tech
Okay.
for two
Hmm.
years, and then I went to graduate school to get my PhD. So you can actually skip straight into a PhD program. You don't have to get a master's
Hmm.
degree.
Oh.
And it was during my PhD program. So that I would say was more of a rough experience, not so much unexpected results.
Hmm.
Again, because the zebrafish were very sick, and the water system that they live in is interconnected, so all of the tanks are connected. So
Hmm.
if there's some sort of contamination or parasitic infection or bacteria, it kind of just spreads to every animal,
Right.
and it's
Hmm.
really, really hard to figure out what it is and to then treat. So
Right.
there, it was much more of a like just a rough experience in a general sense, more
Hmm.
so than unexpected results. Um, right now, during my postdoc, I'm working with mice again. So I've worked with
Hmm.
mice in the past,
Hmm.
and thankfully, the mice are healthy. They're doing fine.
Hmm.
Um, I'm focused on slice electrophysiology. So it's a little
Hmm.
bit different to what I was doing when I was in grad school.
Hmm.
And so what we do is we make slices of the mouse cortex.
Hmm.
And I'm currently working in the somatosensory cortex because
Hmm.
in Fragile X syndrome, one of the common features is sensory hypersensitivity. So
Hmm.
human patients that are affected by this can be hypersensitive to noise,
Hmm.
really bright lights, certain textures. That can
Hmm.
include like food. like food textures may be bothersome to some individuals. It can be like tags on clothing, themes, things
Hmm.
like that. So individuals seem to be hypersensitive to these things. And then they are also, so our mouse model is really prone to having what we call audiogenic seizures. So when
Hmm.
they're exposed to sounds above a certain decibel, they have spontaneous seizures.
Hmm.
Um, so essentially, because of all of that, we're looking in the somatosensory cortex, which contributes
Hmm.
to all of these um, phenotypes that we see.
Right.
And so it's a little bit complicated, but basically, there are a lot of different systems that are involved
Hmm.
in regulating this, the brain activity or
Hmm.
the development of these brain circuits.
Hmm.
And so I'm looking at estrogen receptor alpha, and I'm also looking at endocannabinoids.
Right.
And one of the surprising things is that there are differences
Hmm.
in the receptors that kind of
Hmm.
lead to the same outcome in males and females in
Right.
terms of we see a hyperexcitable circuit,
Hmm.
or the brain activity is enhanced.
Right.
Um, but the way that we're getting there is very different in males and females. So we're still
Hmm.
trying to work out why that is or how that is in males and females.
Hmm.
Um, but yeah, that's really interesting. And another thing that that can lead to is that drug targets might be different in
Hmm.
males and females. So it's really
Hmm.
important to us to figure all of these things out because that can lead to better patient outcomes, higher specificity of medications. We don't want to
Hmm.
give patients medications if we know that they're not going to be effective for them.
Hmm. Right. I think this is a very important project because this can like help streamline which is which and which medication can like apply to others. Um, how do you think that the current field of neuroscience will change over the five or decades or 5 years or so?
Um, I think one of the biggest things that happens in science in general is that we see like really big waves of things
Hmm.
being kind of hot topics to study.
Hmm.
Um, I think that like with the advent of new technology, that always changes how science is done. And I think like one of the things that we're going to see is, and we've already seen this a lot, um, you know, whole genome sequencing was a
Right.
really, really big advent, and that was really expensive and inaccessible to a lot of labs when it first came around, and it became a
Hmm.
lot more accessible over the years. So I
Right.
think that's something we're going to continue to see is that as new technological advances come out, they will become more accessible to more and more labs. I think like single cell sequencing is going to be one of those.
Right.
It's still not super widely accessible, but I think as
Hmm.
the years go on, it is going to become more accessible. And I
Hmm.
think in neuroscience in general, a lot of the things that are going to happen is just that, with the advent of new technology, it allows us to gain more information
Hmm.
or a greater depth of information.
Hmm.
Um, I don't know, it's kind of hard to predict other things in
Yeah.
general. I think
Yeah.
like knowledge is always evolving.
Hmm.
So a lot of the time, people are kind of maybe skeptical of a particular idea, but then as we have
Hmm.
more tools that are developed for us to study certain questions or answer certain questions, then, we may become more convinced of certain things or less convinced of others.
Right. Yeah. And regarding your project that you just mentioned earlier, do you think that this is mostly hands-on work, or do you think that AI will have any influence over this? Or is this mostly about human work and more like manual, like laboratory experience and stuff?
Yeah, so with electrophysiology, I think that's a really important question to ask. I think that the human aspect of the work is going to continue to be predominant.
Hmm.
Um, the only thing that I could say that, maybe AI would be, used in or AI might play a bigger role in is with regards to data analysis.
Hmm.
Kind of streamlining the way that we analyze some of our data. But even then, I don't think that AI will necessarily play a really big role. So
Hmm.
right now, our lab is part of an NIH center group. So
Right.
basically, it's groups of scientists from across the US. We all work on Fragile X syndrome in
Hmm.
some way, shape, or form. And
Hmm.
um, part of our group is comprised of scientists that are really like, more into the bioinformatics aspect of things. And so
Right.
they develop their own pipelines to analyze data. I
Hmm.
don't think they're heavily dependent on AI because a lot of these
Hmm.
scientists are trained as bioinformaticians.
Right.
But with respect to all of the work that I do in terms of the electrophysiology, it is very hands-on.
Hmm.
We have to, obviously, we are the ones raising the mice. We are the ones who are having to extract the brains.
Hmm.
We have to
Right.
prepare the slices using a
Hmm.
vibrotome. We're having to do a lot of work to make sure that the slices stay alive throughout.
Hmm.
It's about a 5 hour long experiment,
Hmm.
and the recovery time for the brain slices is about two hours
Hmm.
long.
Right.
So there's a lot that goes into keeping tissue alive.
Hmm.
Um, and then we're also doing the recording. So we're placing electrodes in very specific areas of the brain
Hmm.
and acquiring information. So,
Right.
I think like maybe the data acquisition itself with respect to maybe the programs that are involved in capturing the data or analyzing the data,
Hmm.
maybe AI will play a role. I'm not, as a scientist, I know some scientists are very on the AI train.
Right. Right.
I'm not so much
Hmm.
for multiple different reasons, but I think it can be useful to some people. I don't think it'll play a really big role in
Yeah.
the work that I'm doing.
Hmm. In this project, how much collaboration does it require? Does it only require specific neuroscientists, or does it require things that go beyond neuroscience or psychology? Because I know you like this is a laboratory based like experiment, but I'm just kind of wondering if you have other experts or other researchers from different fields work on the current project.
Yeah, so right now we're not collaborating with anybody that's outside of neuroscience with respect
Hmm.
to my project itself. That's not to say that we don't collaborate with other groups who study different processes or genes that are not necessarily autism risk genes.
Hmm.
Um, so we've collaborated with a group that's more involved in metabolism and physiology.
Hmm.
And the gene that they were studying is not directly implicated in autism spectrum disorder, but
Hmm.
because of the fact that it's involved in metabolism, they were really interested to see if we could find any changes in the electrophysiology in the brain.
Hmm.
And so another PhD scientist in our lab did some glucose sensor experiments with them,
Right.
and I recorded some electrophysiological traces for them from these brain slices.
Hmm.
So we are open to working with other scientists who don't necessarily study autism or autism related genes, or even, we're open to working with people who study like neurodegeneration,
Hmm.
neurodevelopment, or just any genes that might impact the function of the brain.
Hmm. Have you ran into any specific like roadblocks, or is progress currently smooth?
As of now, progress is mostly smooth. I guess
Hmm.
some of the roadblocks that I've run into is I've done a lot of pharmacological experiments. So
Hmm.
basically, um, again, like I mentioned earlier, part of our goal is to ultimately take this information that we learn and see how we can apply it. We don't do any human patient experiments, and we're
Right.
not directly involved in working with patients, but
Hmm.
the basic science questions that we answer can help other scientists who do work with humans and do, you know, patient trials. It can
Hmm.
help kind of narrow down which drugs are best to use for which maybe like age groups, or if they work better for the different sexes. And
Hmm.
so um, I have tested a lot of pharmacological agents, and sometimes you test those and you get very inconsistent results.
Right.
And we don't necessarily understand why. Maybe the way that they act in the body is different. Um, so whenever we do drug treatments, we call them, I've been doing what's called acute treatment. So
Hmm.
basically, the brain slices are only exposed to the drugs for about 2 hours.
Hmm.
I haven't done any chronic treatments where,
Hmm.
I'm injecting the mice with the actual drug over a period of
Right.
days.
Hmm.
So that might play into it, as to why we get unexpected results. Um, Development is also really important. Maybe the age at which we administer a particular drug or expose the brain slices to a drug is going
Hmm.
to differentially impact kind of the result, right?
Hmm.
So
Yeah.
that's something we always have to keep in mind. So, what the ability of the drug to kind of penetrate the brain tissue and get into all of the cells, that's another thing. So, yeah, that's one of the more challenging parts, I think, is whenever
Right.
you do work with drugs and they're all supposed to be targeting the same thing, but then you get inconsistent outcomes. But the
Right.
way that we balance that is by doing genetic studies.
Hmm.
So, using lines that genetically reduce the expression of a
Hmm.
receptor that we're interested in or a particular
Hmm.
gene that we're interested in.
Hmm. And regarding what you said about the drugs, do you think that if you were to like just theoretically, if you were to put this entire project into like broadcasting for the entire public, do you think that this would have what the public would misunderstand, like what currently happens in your project that the public might call as strange or anything, as like with the context of broader neuroscience?
Can you expand on the question a little bit more?
Yeah. Do you think that the public will, or rather say, what is like the most misunderstood aspect of neuroscience? Like,
Um, so neuroscience itself, I can't really speak to, but I think for me, because I work on autism spectrum disorder,
hmm.
um, and this is a really, really big area of debate for the general public,
Right,
is a lot of people believe that the causes of autism spectrum disorder are related to vaccines. That's
right.
the biggest thing that I encounter as a scientist who does work on autism spectrum disorder.
Hmm.
And so that's always the biggest challenge that I run into when I try to explain to people my science.
Right.
And, a really big part of that is because of misinformation and kind of corruption from a medical doctor that led to
Right.
all of this. And
Hmm.
so I would say that's the most challenging part and also the biggest misunderstanding in the field of autism research.
Hmm.
Um, Especially like I work on Fragile X syndrome. So Fragile X syndrome is actually the number one leading. So basically, it's the number one leading cause of autism spectrum disorder that has a singular genetic cause. So it's
Hmm.
one single gene, a mutation in that gene is enough to cause autism spectrum disorder, and it's the most highly associated genetic mutation that's directly linked to ASD.
Right.
Um,
And oh, I'm sorry. In this workplace, do you think because you are working with autism spectrum disorder and because there are like so many professionals, like national professionals that are trying to solve some problems, do you think they might spread some misinformation as well? Like say if like lab results in their environment were false or something and they try to spread it. Has this happened specifically before?
so I think this is a problem not just in my area of study in
Hmm.
particular, but it's a potential problem in science in general.
Hmm.
I think that the majority of scientists we're usually working towards being as honest as possible, as transparent as
Right.
possible with the work that we do. The reason why when we go through the peer reviewing process before publishing a paper and the reason why it's required is
Right.
so that we can have that rigor in place so that
Right.
other scientists can review the work and make sure that this work is really solid
Right.
and it adheres to the standards. But unfortunately, sometimes does happen where we have people who, will falsify data or, they will um, maybe not necessarily falsify the data, but they
Right.
will overstate kind of the outcomes, or
Hmm.
they will maybe add to their data set in order to make it look like it's more significant than it is.
Right.
That does happen. You know, academic misconduct is an issue, but I don't think it's as much of an issue
Right.
because we have, safeguards kind of in place to prevent that. But it does happen,
Hmm.
unfortunately.
Hmm.
And I think that's true for all professions.
Right. Um, have you ever mentored specific students, or do you plan on mentoring like current graduate students who aim to delve into this field, specifically like Fragile X syndrome or anything related to what you do?
Yeah, so when I was actually in my PhD program,
Hmm.
I taught for 5.5 years.
Hmm.
So one of the courses that I taught for the longest was the cell and molecular biology lab.
Hmm.
So I did work very closely with junior and senior undergraduate students.
Hmm.
And I've taught, some other courses as well. Like I've taught anatomy and physiology.
Right.
Um, I've taught introductory biology. And then in the actual lab itself, we did have undergrads that were working in our research lab when I was in
Hmm.
grad school.
Hmm.
And I have mentored students both kind of over a short term and a long term.
Right.
And I don't necessarily know if they did end up continuing on to pursue a graduate degree or not, but
Hmm.
I always strive to communicate both with my students in a classroom or in a lab setting, um, you know, how to access this career and what the requirements are to get
Right.
toward, to get to be, in graduate school, pursue a PhD.
Right.
So that was one of the things that I always really emphasized to my students. If they ever had questions about this particular career path, to
Hmm.
ask, because a lot of the times we know information or we can direct them towards other resources that can help them become scientists.
Right. And one final follow-up question.
Mhm.
How do you think that this work can change the future for people specifically with this disorder, or rather this disability?
So for me, and I know that, you know, other scientists have kind of their own personal goals related to this. For me, one of my biggest goals is contributing to work that can help improve the quality of life of patients in general. I
Hmm.
think that's what we're always striving towards, at least I am.
Hmm.
Um, I also really care about patient advocacy. I know that um, patients that are impacted by autism spectrum disorder may not always receive the best standard of care. There are
Hmm.
also barriers, especially to individuals that come from um, racial and ethnic minority communities.
Right.
There's a lot of stigma. So I'm Middle Eastern. I know that
Hmm.
autism spectrum disorder is really highly stigmatized within my own community.
Right.
And I think that that's a really big barrier for individuals that are impacted by ASD to get early care, because I think early
Hmm.
care gives them the best outcomes as humanly possible for their future. So I think my goals are always, as a scientist, I feel a responsibility to do more outreach and education towards the general public,
Hmm.
especially marginalized communities. And my goal is always for the work to ultimately lead to something that will help improve patient quality of life.
Right. Thank you so much today for being
Yeah,
with us. I really enjoyed hearing about your project and uh, how your involvement in autism can help the awareness. Thank you very much today. And
yeah, of
is
course.
there anything that you would like to help expand on, or do you think that this broader focus is currently your ultimate task?
Um, so right now, I think I'm really focused on this question.
Hmm.
Um, I'll probably work on this for the foreseeable future.
Hmm.
Um, but my goal during my postdoc, so I am still considered kind of I'm in a trainee phase. So
Hmm.
my PI or the primary investigator, basically the PhD scientist who runs our lab,
Hmm.
she's very interested in these specific questions and how they pertain to Fragile X syndrome.
Hmm.
Because I would like to continue in academia and stay kind of in doing academic research,
Hmm.
part of the postdoctoral training is to kind of push me to start thinking about other ideas and directions that I want to take my own science.
Hmm.
Because ultimately, I would like to establish my own research lab.
Hmm.
And so I should start thinking about some of these questions in more depth and like developing where I want to go with my own science. Because
Hmm.
ultimately, once I leave a postdoc, when I'm kind of looking for a faculty job, that's that's what we have to do is we have to get up in front of a room of other scientists and outline, like what we see ourselves doing in the next 5 years, what's our plan, what research questions are we going to ask and address, and
Hmm.
then an even longer term plan, what does 2 years look like, what does 5 years look like, what does 10 years look like.
Right.
So that's something I'll start thinking about soon, but I haven't even
Hmm.
finished my second year as a
Right.
postdoc. So yeah, that'll be maybe in the next couple of years, maybe years 3 to 5.
Hmm. Thank you for being here. I really enjoyed how your scope and like your involvement in autism has like future involvement. And I think you have like this scope of the project that you can do with which is like very inspirational and impactful to the community. Thank you.
Thank you. Yeah, if you have any more questions, feel free to ask, or,
Hmm.
if you think of anything later on, feel free to email me and I'm more than happy to answer.
Hmm. Thank you.
Yeah, of course. Thanks.